Hi, welcome to IQ Classroom. Today our topic is one of the receptor that is GPCR. Now it's a topic from cell signaling. So today we are going to discuss about GPCR that is G protein coupler receptor. But before discussing about the GPCR, we should understand what is an ligand. Now, as I have written the definition, that ligand is basically a chemical messenger that will set a communication between two different cells, or we can say two same cells. So for a GPCR, we can have different ligands, and we will talk about all those ligands. So ligand is going to, we can say, activate this receptor, and then there will be a conformational change, and likewise there will be a downstream, or we can say downstream signaling. So let's start what is GPCR. First, we will look how GPCR will look like. In a GPCR, basically, if we talk about GPCR, we can see that GPCR is a seven pass transmembrane protein. So GPCR is a receptor that is basically 7 pass. So you can see from the diagram we have here a 7 pass transmembrane protein. We call it as GPCR. GPCR has basically the three domains. The first domain that is extracellular. The second that is transmembrane, or we call it as hydrophobic domain. And the third domain that is cytosolic. In the cytosolic domain, we have three loops, that's L1, L2, and L3. So first of all, we have here the three domains, and we have here the 7 pass transmembrane protein. Apart from that, we have a protein coupled with it. We call it as G protein. So this is G protein, which is basically a heterotrimer that's made up of three subunits, alpha, beta, and gamma. And we have an effector molecule. This effector molecule is basically an enzyme. It can be adenyl cyclase, guanyl cyclase, or it can be PLC. So first of all, we have here the first domain, that's hydrophobic domain. Now this hydrophobic domain, we can also call it as the transmembrane domain. The transmembrane domain. So this is present within the membrane. And this hydrophobic domain, it is made up of seven, we can say, helical structures. There are seven helical structures. So that's why we call it as serpentine, or sometimes we call it as heptahelical. Heptahelical, because there are completely seven passes, seven transmembrane helical structures. We call it as serpentine or heptahelical. Now, extracellular domain, we also call it as amino terminal. Amino terminal. Remember that it acts as the ligand binding site. So this domain will act as ligand binding site. Then we have cytosolic domain, or we call it as carboxy terminal. We call it as carboxy terminal. Remember, as I have already told you, that there are three loops, L1, L2, and L3, and among all these loops, L3 is the largest loop. This is the largest one. Now see, we have talked about the three domains of the GPCR. But in GPCR, we have a G protein. Let's discuss a little bit about the G protein. Remember that G protein is a peripheral protein. So it is present on the periphery. That means it is membrane bound. But if I make a membrane here like this, so this is cytosolic side, and on the cytosolic side, this G protein is present and it is attached with the membrane. Remember, as I told you, that it's a heterotrimer. So that means it has three subunits, alpha, beta, and gamma. With the different molecular weight, alpha has the molecular weight of 45 kilodalton. So that means this is the largest one. Then if we talk about the beta subunit, the molecular weight is 37 kilodalton. Similarly, for gamma, the molecular weight is only 9 kilodalton. So that means this is the smallest subunit. One important thing I am going to mention here is that we have three subunits here, that is alpha, gamma and beta. And at the same time we have told you that G protein is attached with the membrane, and it is attached with the membrane with alpha subunit and gamma subunit, not by beta subunit. So beta subunit is not attached with the membrane. Now listen, we have three subunits here, that is alpha. We also denote it by G alpha because it's the alpha subunit of the G protein. We represent the beta subunit as G beta, and we represent the gamma subunit as G gamma. Remember, all the three subunits remain attached with each other when G alpha is attached with the GDP. So when G alpha is attached with GDP, it has higher affinity for beta and gamma, so all the three subunits will remain attached until the GDP is attached with the alpha subunit. But once GDP is replaced by GTP, the G alpha gets separated from beta and gamma subunit. Now, apart from G protein, we have here an effector molecule. As I told you that it is plasma membrane bound enzyme, and we have here examples. It can be adenyl cyclase, it can be guanylyl cyclase, or it can be PLC. So we have different effector molecules. So let's see how GPCR is going to work. Listen, in GPCR functioning, the first thing that's going to happen is that the ligand will come and bind with the GPCR at its binding site. Once it will bind, it will lead to the conformational change in the receptor. Now this conformational change, due to this conformational change, the L3 gets activated and it gets attached with the alpha subunit of the G protein. Once L3 gets attached with the G alpha will sense this conformational change and it will replace the GDP by GTP. And once GDP is replaced by GTP, the G alpha will lose its affinity with the beta and gamma subunit and it gets separated from beta and gamma. Now this separated G alpha is going to activate the effector molecule. Say for example, in our case it is adenyl cyclase. Now this adenyl cyclase, upon activation, will convert ATP into the cyclic AMP. Now cyclic AMP will act the secondary messenger. It will act as secondary messenger. Now this cyclic AMP will recruit the protein kinase E, and it's also going to activate, we can say the cyclic AMP is going to recruit protein kinase E. This protein kinase E will do two functions. Number first, it will phosphorylate certain cytoplasmic proteins, which in turn will activate other enzymes or proteins so that there is a response. On the other hand, this pKa can directly activate the transcription factors like we have CRAB, we have CBP, so that there will be a response. So this is GPCR functioning. First the ligand will bind, there will be conformational change that will make the activation of the G alpha. G alpha will activate the adenyl cyclase or any other effector molecule, which in turn will activate or lead to the formation of its secondary messenger. So we can see here different effector molecules have different secondary messengers, and different secondary messengers will activate the different proteins. Like I have framed it in the tabular form. You can see for adenyl cyclase we have the secondary messenger cyclic AMP, and the protein it's going to activate is protein kinase E. Similarly, for guanylyl cyclase we have the secondary messenger, that is cyclic GMP, and the protein it's going to activate is PKG. For PLC, the secondary messenger will be IP3 and the DAG. Now here IP3 is going to help to release the calcium dipositive, and similarly DAG will recruit the PKC, which will do the further functions. So In the next video, we will understand one of the application of the GPCR, that's a mechanism of glucagon. Stay tuned. Thank you.